Hello, um, I'm I'm Jean-Luc Legras, and I'm at the head of the Serum Levure Yeast Collection at INRAE, and we have organized this webinar in the frame of the European project MIRRI 21. Sorry, I would like all of you to welcome you for today's webinar. And why this webinar? We can say today that microbial resource centers are at the crossroad as they face at the same time two major challenges: the microbiome revolution and as well the arrival of the massive sequencing area. So we will have to answer how we as MIRRI we are going to cope with these challenges. Today's webinar is divided in 3 sessions. This morning, speakers will present their approach and results in order to cope with the issues of microbiota conservation, and then culturomics methods for the isolation of microorganisms. And in the beginning of the afternoon, we'll be presented the use of MALDI-TOF MS and new tools for the analysis of MALDI-TOF data. And in the mid-afternoon, we'll be presented some more examples of the use of genomics for taxonomy. So, and along the sessions, Francisco Rocha and then Marta Avromova will collect your questions, and at the end of each talk there will be a 5-minute short time to try to answer your question. But please do not hesitate to write this question in advance so that we could more smoothly present them. And at the end of these 3 sessions, there will be a time for exchanges between all the speakers and with the audience. So to start with the microbiota session, I'm very pleased to welcome our first speaker for today, who is Dr. Matthew Ryan. Dr. Matthew Ryan is the research lead for biological resource and curator of CABI's Microbial Culture Collection in UK. And I have to remind that CABI was one of the initial partners that participated to the creation of the MIRRI project some years ago. And Matthew will present how CABI addresses the challenges raised by microbiome to microbial resource centers. I'm sorry. Please, Matthew, the floor is yours.
Thank you very much. And I've now lost my PowerPoint completely, which is an absolutely brilliant start to the morning. So please bear with me. It's the effect of Boris Johnson and the English Channel, I guess. So many apologies. Just bear with me. I'm going to try and reload it. Sometimes we cannot beat the technical issues. I apologize. Okay, can you see that?
Yes, Matthew.
Right, brilliant. Thank you very much. So thanks for the introduction, Jean-Luc, and it's an absolute pleasure to meet and talk to the MIRRI family once again. As Jean-Luc said, that we were involved in some of the very earliest meetings in Brussels and Braga regarding MIRRI, but unfortunately we weren't able to take part in the main project, and it's exciting to see it developing. My talk this morning is about culture collections and the microbiome, challenges and opportunities. It is loosely based on a talk that I gave to Echo 39, but I'm going to tease out some of the more interesting aspects that relate to the research infrastructures that we have across Europe and also the international scene. And I'll do that by looking at the current status of culture collections, address the microbiome challenge, and then with our exemplar project that we have in the UK, which is a UK crop microbiome project, which is bringing some of the different aspects of microbiome R&D together. And then I'll look at some of the challenges and opportunities that we all face as culture collection scientists and how we meet the needs of our communities. But at the outset, one of the most important considerations is what is in a definition, because the field of microbiology, it's, for some people, it's not just culture collections. For us, these are the collections of exenic microbes, be they algae, bacteria, virus, animal cells, or even algae, and the associated provenance data. But the biobanking community is also very strong. Some collections even describe themselves as biobanks. They tend to be mainly medical and human veterinary orientated, but they do develop into other areas, and it can be quite it can act as a mechanism of competition between the two communities. And then we have a term that we're all familiar with in MIRRI and beyond, and that is the OECD definition of a biological resource centre. And that is really where I think we need to aim for with respects to microbiome-based collections. And they are the part of the infrastructure that underpins biotechnology. But of course, there's also cryobanks, MDRCs, DNA banks, MRSCs, and others. Collections in the traditional sense are still an absolute treasure trove of useful cultures. To our surprise, we held the Fleming strain for many years, and it wasn't fully sequenced.
Excuse me, Matthew. Are you still at the very first slide?
No, I'm not. I'm on slide 4.
Unfortunately, because right now I just don't need the slide 4 arrive. You are just we have skipped the 2 first ones. So
okay, can I suggest that you share the slides I did yesterday, because there's definitely gremlins in the system this morning. Is that possible from?
Thank you, Alexandra. So we are back at the first slide.
Yeah, if you could go to slide 4 then, please.
You can change the slides by yourself. It's still working. With the arrow on your laptop.
Yeah, okay.
Can you try? No? Okay, then I'll do it for you. Oh, yeah. Okay, perfect.
Yeah, I've got it. Right, thank you. I just got to get to the right side. I can't see a full screen, though, at all here. Not ideal. Okay, so yes, as I was saying, the collections have a treasure trove of microbial resources that we all have in our holdings. We had the penicillium strain, but collections across the EU and globally have lots of useful cultures that are still relevant to science. And the onset of genomics and bioinformatics has really helped develop how we exploit the resources that we hold in our collections. But as we know, cultures aren't solely stored in exenic form. And in the environment, a culture is there, a strain is there with many hundreds and thousands of other strains. And through the EU Microbiome Support Project, we've begun to look at the broader picture. And this is a lovely schematic that was produced through that particular community support action, really addressing that human and animal microbiomes are prioritized occasionally over the environmental and plant associated microorganisms. We can take this further by looking at the whole system, or as we describe it as the phytobiome. Others may call this the holobiont, but it's where you have a plant and the microbes in association with it. But it impacts on all the other organisms within the system, whether they are insects, worms, birds, or other mammals. It's about what impacts on the microbiome system, the climate and the weather. And so, yes, it's a very important approach to our conceptual understanding of how microorganisms act and communicate with other within a particular environment. But what about current collections? There is no coordinated strategy to preserve microbiome samples currently. We have the traditional collections such as CABI, Dsmz, the Westerdijk, Institut Pasteur. We have another tranche, which is the medical and healthcare biobanks. which, as I said previously, tend to be more medical, but they can include other resources as well. And then we have the environmental collections, such as the seed banks. One such example in the UK is the Millennium Seed Bank, which is a really fantastic resource of seeds. But associated with those seeds are bacterial and fungal endophytes. However, when the seeds are preserved, the focus is on preparing the seed for cryopreservation and not the fungus or the bacterium associated with it. And this can be a brilliant resource for future research and development. So looking at it from a top-level view, again, this is an output from the EU Microbiome Support Project. We looked at the infrastructures supporting microbiome. And what is interesting is that, again, as we see on the level of the biobanks and the collections, the infrastructures are in fact very fragmented. A microbiome tends to fall in the gap. We've got the collection-based networks, such as the World Federation of Culture Collections, such as MIRRI, the US Culture Collection Network, for example. But we are a community that generally we look after ourselves. Then we've got the biobanking community, an organization such as ISBA, which is a global organization, as we know. And then you have the data and bioinformatics expertise, and they form their own consortium and infrastructure as well, such as Elixir and EBI. And then, just to make it even more confusing, there's all the other projects going on in Europe and around the world that might or might not involve the use of microorganisms, whether it's EMBRIC or Corbel or many of these other what was Horizon 2020 projects, of course, soon to be Horizon Europe. And this will continue. But again, the emphasis here is how can we, as a research community, bring this all together to support this critical area of research that is actually being dictated to us by the very user community that we serve. And they are the research microbiologists across Europe and across the world. An output of the EU Microbiome Support Project was our paper that appeared earlier this year. Nelson Lima was a co-author on this, and that was Development of Microbiome Biobanks, Challenges and Opportunities, where we addressed some of these issues and asked ourselves some really pertinent questions about how we should support this area of research. Because it's not just the infrastructure, it's also the science behind it. It's the data, it's the bioinformatic analysis. And that is really important that we don't lose track of any aspects of that. But the requirements for microbiome biobanks are similar to what we do for culture collections at the moment. It's to aid the development of standards. It allows deposits in compliance with legislation, including the Nagoya Protocol and deposits under the Budapest Treaty. And that is an area of real concern because there are very, very few culture collections that will preserve and undertake to successfully ensure the viability of any microbial consortia. Industry demands that we do this, but as a community, we're not there and the science isn't quite there either. Biodiversity conservation is another area that we often ignore. Biodiversity is really high in the media's attention at the moment with COP. But as collections, we don't tend to just go and get an environmental samples and store them for the benefit of humankind. Yes, that takes a lot of resources, but maybe a coordinated approach, particularly within climate change, is warranted. And ultimately, as with all resource provision, it's about ensuring the quality and stringency of the very research we do. And as we knew with the EMBARK project many years ago, it was about getting scientists to deposit their strains in culture collections so we could repeat the work. On a country level, different nations are applying their own approaches. In the UK, we have the Knowledge Transfer Network, and they produced a microbiome roadmap. This brought together all of the different communities, from the humans to the veterinary, from industry to research academics. And as you see, right in the middle of these key priorities is microbiome standards and biobanking. And I have an opportunity within the UK to help now lead a strategy for ensuring this community is underpinned. And this roadmap is available online. If you just Google KTN microbiome roadmap, it's there for everybody to see. But we're culture collections, and some of our colleagues will take fright at the prospect of having to handle really complex samples. And so some of the questions that get asked, haven't existing collections got enough to do? It's not our problem, leave it to somebody else. Let us just focus on the xenic organisms. The other aspect is the business model. The business model we have for culture collections will not work for microbiome collections. The prime reason for that is that sometimes the resource is finite. You might only have 2 or 3 samples. You can't always take the cultures and reprocess them, depending on what approach you're using. But where is the science behind all of this? And this is another really important consideration. Cryobiology is the science behind all of our low temperature preservation regimes. But actually, there's not a lot of research going on in collections across Europe. The reason for this is, of course, if you're working with mixed samples, we know that the cryopreservation procedure and the stresses that we encounter during freezing and cooling will actually kill a vast amount of the organisms within a mixed sample. So therefore, we have to develop methodologies that restrict this damage. And then there's a question of functional potential, which is really important as well. It's okay preserving something, but what's it like when it comes out at the end? Has it still got the potential to undertake a biosynthesis reaction or something similar? These are all really important. And have we maintained the genomic integrity of those preserved samples? As we've seen with many other areas, there's a lot of work going on in the human gut area. And sometimes this translates to the environmental and crop health areas. And in the UK, we have started to do something about that. And we've been very fortunate to have gained funding to start a UK crop microbiome project. It's worth £2 million. And what it intends to do is bring together biological resources, cryopreservation expertise, crop health, microbial ecology, and bioinformatics in order to deliver a microbiome genetic resource-based project. So why did we set up the project? Well, we knew that existing culture collections didn't really support microbiome research. It was driven by the needs of industry, and that is big industry as well. We responded to the need to link samples and their metadata. Vincent at the Westerdijk has been a proponent of this over the years. It's really important not to lose a connection between the sample, its provenance, and its data. But as we all know, when you go onto GenBank, if you find an interesting sequence, the chances are you won't find or even be able to access the original sample. And again, another reason, there is no standards underpinning microbiome research, or very few. There are moves with various infrastructures around the world to now do this, particularly from the data perspective. And for our project, it was about looking at the major crops. So our intention was to establish a cryopreserved and characterized crop microbiome resource to underpin UK and international crop research. We're focusing on key crops. We have to set a baseline. We can't do everything. And that's barley, oats, oilseed rape, potato, sugar beet, and wheat. And we also have a bean crop. Our ultimate aim is to provide that comprehensive platform to facilitate research towards optimizing plant yield in an integrated crop management framework. So it's very focused, but is incredibly important because we have to meet the UN's Sustainable Development Goals, and this is just a very small part of being able to do that. Our key aims are to provide a validated resource for researchers, to source data, to be able to repeat their work, and to provide them with living material. And because of this, we're using two approaches. We're using a culture-dependent approach. Some people might call that culturomics, and we're actually preserving soil samples and plant material. And this is how it all fits together. CABI's activities relate to the cryobank itself, which is coloured green on this schematic. But all partners, and it's a multi-partner project, have their own specific areas, and it's a real good example of collaborative research. We have Rothamsted doing the culturing, obtaining the samples. We have colleagues at the James Hutton's Institute and Scottish Rural College doing our bioinformatics. And then the John Innes Institute are helping to characterise the collection with a view to providing products for industry. And this is all linked in with a new database, which I will come tell you about in a short while. The project consists of 4 work packages. The first one by Rothamsted, and they will be undertaking the task description of getting the soil, growing cultures, growing plants to see what microbes the plants accumulate, and again focusing on those key crops. They'll also be extracting the DNA at a very early stage, so we will also be having an approach to bank that DNA. We're utilising Rothamsted's participation in the ASSIST project to get soils from across the UK. So what would be great is if we could do this on a European basis as well. So there's a challenge for MIRRI and also other research collaborators. So we've got a very broad range of soil, and then we take the soils, they prepare the soils, and importantly, they get all the metadata involving those soils, whether it's GPS and location, right through to pH, phosphate concentration, and all the other physical chemical components, which is really important. So this is a really great data set. This is what the soil looks like. And as you can see from this image, there is an array of different soil types that we're looking at. And you can see that from the color actually. And it's a nice image just summarizing that. And that's in the greenhouse at Rothamsted. And then we grow the plants in the different soils. We collect the soils post-growth to see what the plants have actually accumulated microbially. And that's when the samples are sent to us at CABI and we do the cryopreservation. We're using, as I mentioned, both a culture-dependent and independent approach. And this is important that we capture as much as we can from our samples. And again, that's using a culturomics-based approach. We're also using high-throughput approaches. So we're preserving in 96 well plates. That does present problems because, of course, if you want just one sample, you have to defrost the whole plate. But these are just some of the teething problems that we're coming across. The reason for that is our cooler at CABI can't handle anything more than plates that don't have the individual wells that you can actually remove. They're often called omniplates. Our partners in Scotland are doing all the bioinformatics and genomics, both for bacteria, which is following a standard 16S approach, but also the fungi, and which we're using an ITS based approach. But again, these are standard metagenomic based approaches that we're using. And I won't dwell too much, but this is a standard workflow that many of us today are familiar with. One of the things that we are doing is we are putting this information and making it publicly available through our project website called Ag Microbiome Base, which is a curated and open access ag microbiome base of the bioinformatic information, the metadata, the annotated sequences, and we're linking it actually into EBI's Magnify, which of course is a really useful tool that they have and they are developing. It's got a lot of human veterinary information on it, but we're going to be updating our environmental information to it. So that is a really, really great resource. But again, the most important thing is we're going to link everything from the soil data, from the sample provenance to the organism itself, and right out to that bioinformatic information. All of it is publicly available. And then comes Cavi's work package. We're looking at the cryopreservation. The goal is to conserve and store crop microbiome samples and develop those best practices for doing that. It's quite a large project, and to enable us to do that, we are employing controlled rate cooling technology. I'll just go on to the next slide. And an alginate encapsulation approach. Stirling cycle is an advanced cryopreservation method, which benefits from the fact that you cryopreserve and cool the samples on a plate that's attached to a Stirling cycle cooler. This means you have brilliant reproducibility, and there is very, very little variation across a batch. This is much improved from the traditional chamber coolers that pump cold gas, where the variation in temperature can be quite marked. So if you want to maintain good stringency of your approach for cryopreservation, that is something that it is a very recommended approach. Even if you're doing it for exenic organisms, it's a far more superior way of cooling samples down to ultra low temperatures. We're also working on an encapsulation dehydration technique, which was originally developed through the EU COBRA project, which was an algae project. It was then developed by the EMBARK consortium way back with CECT, Minnow, and the Westerdijk Institute. And it's great that these technologies that we develop have application going forward. It really shows the value of that European-based collaborative research. And so far, we're making good progress. We've already preserved 350 bulk soils and have 350 encapsulated soil cryotubes. We are looking at working on our standards, and we're hoping to, but we actually have published a paper through AgriArchive, which will be submitted to a journal shortly. So it's a very exciting area. Very briefly, I will just move on to the final work package, which is being led by the John Innes Centre. Again, this shows the real importance of collaborative research. So their job is to take the characterized microbial isolates and actually look at them for industrial development. So one of the things we're going to be doing is developing synthetic communities with potential agricultural use. And this is the workflow that they're using at the John Innes Centre. Obviously, we've got the microbiomes. We then do the single isolate collections before the implanter screening. So we'll be using an autobiotic system to look at such things as water stress and nutrient availability, with a view to developing synthetic communities that have direct agricultural application. And this is really important because this is something that industry is demanding of all of us as preservation scientists. So in summary, our key outputs of the project is the cryopreserved resource itself and access to the genomic data. It's the development of those robust methodologies for collection and storage. It's about enhancing the sustainability of those genotypically and phenotypically stable resources. Again, I mentioned functional potential, and that's very important. But it's about providing the added value to the community. And again, we are very passionate about that. The sort of studies that these projects can underpin, this is related to our project, but this could equally be for any other microbiome-based project. Once we've got the samples, there's so many things we can do with them, whether it's understanding population dynamics and their evolution, whether it's the snapshot in time with respect to biodiversity and conservation, where we preserve now, before the impacts of climate change, to help us mitigate solutions as we move forward, whether it's the identification of those functional traits or the impact of gene mobility on the plant microbiome. So in summary, genomics and bioinformatics have revolutionized our approaches to microbiology. Developing technologies and the scientific community have demonstrated the need for collection and biobanking capacity to support microbiome R&D across Europe through the EU Microbiome Support Project and globally as well. We need to develop the infrastructure to underpin that research. We know it's fragmented at both a national, regional and international level. Microbiome-based methods can't fall between the gap. So there needs to be a concerted effort from those communities to work together, whether that's MIRRI, ECHO, EU Microbiome Support, EBI. No one in infrastructure can do it alone. And that's why it's important that we can come together and work collaboratively to really develop and meet the needs of the microbial resource community. And another consideration is also the One Health aspects. Time and time again, we tend to find communities work on their own, whether it's the plant and crop community, whether it's the veterinary community, or whether it's the healthcare community. By coming together, we can learn off each other to develop the solutions that we all need and translate that research into achievable outputs. And that is where the opportunities are. Microbiome research demands collaborative R&D at whatever level, whether it's the researcher level right up to the infrastructure level. And again, that's where MIRRI has a great opportunity to make its mark. So I'd like to thank all of my collaborators involved in this project. That's my colleagues on the UKRI BBSRC UK Crop Microbiome Cryobank project at Rothamsted, the John Innes Centre, Scottish Rural College and the James Hutton Institute, but also some of the background work that I've presented today through the absolutely fantastic EU Microbiome Support Project, which is brilliantly led by Angela Setic in Vienna in Austria. Before I finally sign off, I just got a couple of quick slides for anybody interested in microbiome-based approaches. The Phytobiomes Alliance have a conference in person, I'm pleased to say, in Colorado next year, looking at some of the issues we've discussed today. And what this has done, this is really an academic and research collaborative. So if you're interested in this, do go out to that conference. They are absolutely fantastically well-organized events. And then in the UK, we're holding the 3rd International Plant Microbiome Symposium, again in person in Dundee, Scotland, and there will be microbial resource elements to this as we realise it's an essential component to underpin everything. Finally, I'd like to thank John Luke, and I do apologise for the technical issues, but sometimes technology just beats us. Thank you very much.
Thank you very much, Matthew, for this really brilliant talk. It was really a very nice presentation. So, Francisco, I think you maybe have some questions that have been addressed in the forum.
Yes, Jean-Luc, and thank you very much, Matthew, for the excellent presentation. Again, we do have a couple of questions here in our forum. I will just read them. The first is from Ksenia Chekina regarding intellectual property. Can you tell me more about intellectual property regarding the culture collections? What strains are patentable? What strains are not in your collection? And what is the percentage of each group? Also, Ksenia is asking who pays for the maintaining patented and open sources part of the collection. Matthew.
Right. Okay. That's a really complicated collection. It could probably take half an hour to answer because there's so many strands in there. I think for our project, we're making it publicly available. We are loosely following the ECHO material transfer agreement, as all European collections do. So we are making this resource publicly available. If industry comes along and wants something, we will negotiate a commercial agreement to really provide the resource back so we can maintain the collection going forward. The broader question regarding IP protection is really, really important. The Budapest Treaty has limitations about the preservation of consortia, but it is something that the intellectual property community, the patent attorneys, are really demanding of us at the moment. But the science is just not quite there to deliver it yet. We can do and preserve small consortia, but the more components you have in those consortia, the more difficult they are to preserve. So that is a really immense scientific challenge. I think with the developments in high throughput biopreservation technology, we can begin to answer these questions and solve the problems. But this is an area that requires a lot of research attention, and to work collaboratively with MIRRI and partners on this would be absolutely brilliant. Thanks a lot, Matthew.
We do have another question from Nelson. First, a note on the very nice presentation you just gave us, Matthew, again. The question is, do you foresee that microbiomes really could be preserved as specimens, that is limited sample in quantity, to allow during the time the same composition and function? And what kind of standards do we need to prioritize?
Yeah, another great question, Nelson, and I hope all is well in Braga. Yeah, that's really important. So the functional potential is something we're putting a lot of focus on. So when we preserve our samples, we are trying to optimize the techniques as much as we can, because we know they're only a finite resource. So if we've got a soil sample, we've got maybe 10 individual vials of those particular samples. So we don't want to give them out immediately, because that's not great, because then we won't have this ongoing record. So there is an issue about the sustainability of these resources, but we felt it was important to provide that baseline, and that's what we're trying to do with this. Sorry, and what was the other part of the question?
The second part was what kind of standards we can prioritize.
Yeah, absolutely. So we need to look at the cryopreservation standards, but also how collections can provide standards for the validation of techniques in the microbiome research arena. We obviously do this quite frequently now with test strains. I think most collections hold test strains, but of course there's an increasing demand to be able to validate research in the microbiome area, and that is a challenge again, but it's something that can be done. And I know colleagues in other collections throughout Europe and globally are also looking at this, and the EU Microbiome Support Project actually had a session which was led by Michelle Sloter from Munich, and so this is I think Nelson may have been actually part of that, but it's an area that the infrastructures really need to look at.
If I may, I think we have despite the old interest of the question, we have to move forward and keep this question for the discussion session at the end of the. Otherwise, we won't stop basically. So I think we have to move forward. Okay, thank you very much, Ryan, and see you in a few moments for the question and answer global session. So now we are welcoming our second speaker, who is Dr. Thomas Hidel. Thomas Hidel is a product manager of the Dsmz infrastructure, bioresource, biodata, and digital health, and project manager of a Dsmz. I'm sorry for my German, and pathogen repository at the Leibniz Institute at Dsmz. Again, I have to remember also for those, Dsmz was also a partner from the early phases of the construction of MIRRI, so we have not forgotten you. And today, Thomas will report on current development at Dsmz and its Dsmz pathogen repository, and the focus will be on collection of microbiome strains from the gastrointestinal tract of mice, pigs, chicken, and humans, as well as on microbial consortia. Sorry. Please, Thomas, the floor is yours.
Thank you very much for the introduction. I try to share my screen now. Can you see it?
Yes, yes, perfect.
Okay, I try, or I thought to split my talk into two parts. At first, I want to give some overview about the Dsmz for those people who might not familiar with our institute. So the Dsmz is the most diverse biological resource center in the world and one of the largest collection of microorganisms and cell cultures worldwide. And we have approximately 80% of all formally described bacterial species. And the Dsmz hosts the majority of bacterial diversity, as well as globally unique stock of human and animal cell lines, fungal strains, plant viruses, bacteriophages, as well as genomic bacterial DNA for research purposes. And we are both an active collection and a research institute. So we have in our collection around 79,000 items, including around 35,000 different bacterial strains, 80,000 fungal strains, around or nearly 1,000 human and animal cell lines, 5,000 plant viruses and antisera, 1,000 bacteriophages, and we also have in our collection 90,000 different bacterial genomic DNAs. So the Dsmz maintains specific expertise and offers counseling in the areas of bacterial taxonomy, phylogeny, and species description. And we have a standardization and equality assurance of bioresources. Furthermore, we are offering issues in biosafety and biosecurity, and we have a legal framework for exploitation of bioresources regarding patenting, CBD, access and benefit sharing, and as well as the Nagoya Protocol. So we are also active in research, especially in microbial diversity and the underlying evolutionary mechanisms. Then we are active in diversity of microbial functions and adaptations. And we are active in molecular mechanisms of biotic interactions. So the Dsmz employs around 200 people, and we have 70% of women overall. 45% of the employees are scientists, and 50 of them are women. And the proportion of female non-scientific staff is almost 90%. So the Dsmz was growing up in the last years a lot. This is the main building, how it looks like, or how it looked like in 2009. And it was too small and not nice enough. So then the building was getting one floor plus. So that was in 2014, the building process. This was in 2015. And now it's finished, and much bigger and nicer, but it became that it was too small. So this was in 2019, a second building or a part of a second building. And this is now, so the pictures from yesterday in 2021. So we have a part of a second building and the Dsmz is increasing. So some words to the history fastly. So it was founded in 1969 as Deutsche Mikrobenbank in Göttingen. In 1987, it was relocated to Braunschweig. In 1988, it was founded as a non-profit organization with Niedersachsen as a sole shareholder. In 1990, the extension, so the Dsmz extended the collection by human, animal, and plant cell lines, as well as plant viruses. In 1996, it became a member of the Leibniz Association. In 2011, it was renamed into Leibniz Institute Dsmz, German Collection of Microorganisms and Cell Cultures. And in 2018, it became the first European registered collection. So here are some pictures, how it looked like from the beginning to the end. So here you see ampoules or Hungate tubes in the normal fridge or freezer. This is the ampoule storation, how it looks like in the Dsmz at the moment. Here we have also the liquid nitrogen storage as an example. And here you see the newest thing. So some or many ampoules are now stored in robotics. So then a short overview about our diversity. So we have several distinct curatorships here in our collection. So they are connected to microorganisms, bioresources for bioeconomy and health, human animal cell lines, and plant viruses. So microorganisms connected to gram-positive and gram-negative bacteria, archaea and extremophilic bacteria, halophilic and phototrophic bacteria, geomicrobiology related bacteria, underrepresented bacterial phyla, and cyanobacteria and protists. Regarding the bioresources for bioeconomic and health, so we are focusing on pathogenic bacteria connected with the Dsmz pathogen repository, where I show some slides afterwards. Then fungi, fungal systematics and health relevant fungi. Clinical phages, phage-related genomics and application, actinomycetes, and applied microbiology. And regarding the human and animal cell lines, we are focusing on cell diversity, tumor diversity and biology, functional tumor genomics and molecular genetics. And regarding the plant viruses, we are focusing on systematics of plant viruses, molecular plant virology, resistance mechanisms, and diagnostics of plant viruses. So here you see the complete overview in some pictures in one slide. And what are the traditional tasks of the Dsmz? So we are trying to maintain the authentic biology resources, preservation and deposits. And you can shop all items we offer via web shop. We also have a patent and safe depository according Budapest Treaty and IDA, and central services like genomics sequencing, MALDI-TOF, and so on. We are really interested in research and teaching. So here you see the biological diversity in safe hands, how we have it here at the Dsmz. So we have cytophilic, mesophilic, thermophilic, and so on, all what you see here in our collection. And today I'm trying to go more in the pathogenic, non-pathogenic, microbial, microbiome area. So here you see how we cultivate the microorganisms in 2 pictures, really simple. So we have more than 1,500 different media with up to 100 ingredients. And they are splitting they are splitted into ready to use and homemade media. So here you see some insights, how it looks like in our laboratory. So as many of the microbiome gut samples or gut strains are anaerobes, we have to go to anaerobe TENS or Hungate techniques, which we have established here in the Dsmz. So here are some more pictures, how it looks like. And here are some more pictures, how it looks like. And you can split also the cultivation of microorganisms into old school mechanisms or high throughput cultivation. So old school, so you select your media according to your needs. You use different incubation conditions, avoid contamination, and have patience simply. And then how it went into the past. So you have to do high throughput cultivation. So you are verifying or varying your substrate concentrations. You have different substrates, and you are carrying ionic strength buffers and so on. The final goal in both things is to obtain pure cultures. So here are simply shown the preservation techniques, how we have it in the Dsmz. So freeze drying on the one part, liquid nitrogen on the other part, and 80 degree freezing as glycerols in the third part. So now I'm going to the Dsmz pathogen repository, and it's located within the German Center for Infection Research, and it's located in the Dsmz, and we are coordinating the banking of bacterial bioresources through this Dsmz pathogen repository. And here you see the Dsmz partner sites within Germany. So we have Munich, Tübingen, Heidelberg, Gießen, Bonn, Hannover, Braunschweig, and Hamburg, Lübeck, and so on. So the tasks of the Dsmz pathogen repository. So we are offering a deposit free of charge in an open collection, closed collection, safe deposit backup within Dsmz. And we are offering extensive authentication and molecular chemotaxonomic characterization of all strains. There's a regular quality and viability testing, and genomic DNA can directly supply for molecular studies. If wished, genomic sequencing with PacBio long-read sequencing and Illumina short-read sequencing, it's included. And we are offering online documentation and online oral system. And it's all connected with professional and rapid delivery, and all legal conform with Risk Group 2, CBD, and Nagoya Protocol. And we have also access to international collections via network. So the collection highlights of the Dsmz pathogen repository are multi-resistant strains with several antibiotic resistances, then key strains from recent outbreaks of multi-drug resistance bacteria, raw clinical isolates, and key strains from intestinal microbiomes. And I'm trying to focus on this in the last slides. So here you see how the data pathogen repository increased in the last years. So we are harboring 3,000 isolates around in November 2021. Here you see the diversity of bacteria according their number we have in our collection, and we are harboring 21 different genera now. And I think this splitting of different bacteria has also a major or was driven from by several new taxonomic descriptions based on microbiome isolations or strain isolations. So the data pathogen repository is a mix of collection, service, and research. And here you see how it looks like, or how it looked like in the past or over the years until 2020. So the microbiome research, or the number of strains which we have in our collection, increased from 2014 to 2020 a lot. So we have over 500 strains only isolated from microbiomes. So now I'm going shortly to the mouse intestinal bacterial collection. So it includes all cultivate bacterial strains isolated from the gut of mice that are publicly available up to date. So we have in our collection 109 strains, including new genera and new species. And this is or shall be a resource building to increase the knowledge of cultivatable diversity of the mouse gut microbiome and support scientists for colonization experiments in germ-free mice. And you can have a short look if you use this link. So when you use this link, you will find this page, and there are listed 109 different strains isolated from mouse gut microbiomes. So here you see we separated the microbiome into several strains. Then at the moment, there's an extension of the cultivatable bacterial strains isolated from the mouse gut in process. We are working on around 100 additional strains at the moment. So here you see the same thing or a similar thing for the host for a pig. So we established a publicly accessible collection of pig gut bacteria, and it's similar to this of the mice microbiome. And you can have a look, if interested, to this link. And here is a short overview how we did this in collaboration. So you have the pig, and we sampled the fecal things of the pig and put it into 50 milliliter of anoxic media. Then, so it was included. So you have then this little bit oxygen inside. You have to shake it really fast, because as you know, some samples maybe are very or they do not survive little things of oxygens. So then after shaking 1 minute, you take the sample and you try to include it into anoxic media again. And then you afterwards, after growing or within growing, you can split it into Hungate tubes or agar plates to let them grow and survive. So here you see how it looks like when you split the microbiome into parts of the microorganisms. So we were able to comprise 110 species across 40 families and 9 phyla. And inside of that, we had 38 novel taxa, including 29 novel genera and 16 novel species. I try to hurry up. Here you see the page, how it looks like for the picture. And then we also tried to change the host again. Again, the same for the chicken. Here you see the picture or this, the strains we were able to isolate from the chicken gut. The collection comprises 43 species across 19 families and 4 phyla, and we were able to describe several novel taxa, including 3 novel genera and 4 novel species. So then we are at the moment in process to increase the human gut microbiota strains in our collection. So we have already 2 big ones in our collection, but we are in process to increase it. So, and to shortly try to go from the splitting into strains. So we are also in process to put them again together as an artificial microbiome in the same media, ready to use for research in mice. This is an example for mice. So it's an artificial microbiome. This can be used in mice on request, and there we are also very active at the moment. And to show the last slides, and then I'm finished. You can easily take our bacteria for research, and the tasks are to keep them happy, healthy, and well, give them away. And as you see here on the left picture, they can even glow in the dark. So here are my acknowledgments. The people which are mainly involved in our microbiome research, on the one hand in splitting the microbiome into parts, and on the other side putting them together as artificial microbiomes ready to use. So I'm very happy to collaborate with them, and also to all others who are involved. And with this, I thank you for your attention, and I'm happy you can contact me. And if any questions arise, just let me know. Thank you very much for your attention.
Jean-Luc, I believe you are muted.
Okay, thank you very much, Thomas, for this nice presentation. And I give a very nice overview about the activity of the Dsmz and as well of the I'm sorry about the name of your pathogen.
No worries.
DCIF pathogen repository. So basically, I would say, in short, that the strategy of Dsmz to keep on with microbiota is in fact to explore as much as possible the microbiota and isolate as maximum as possible the number of strains in order to rebuild, to be able to give the possibility to rebuild this microbiota. Is that right? Is that what I understood?
Yes, yes.
Yeah. So yes, it's another philosophy in contrast to that of Matthew, this just the former presentation. But it was really I was very impressed in how he developed the tools especially to work with anaerobic bacteria. So do we have any more questions, Francisco? I mean, one will have to be quick, so because it's because the program is pretty tight this morning.
Jean-Luc, we do not have any questions as yet. I don't know
Okay.
if you want to move forward or wait a little
So, okay, I think we should go forward, and we have and if people have new questions, we'll go back forward at the end of the morning. Okay. So thank you again to Thomas, and please do not leave. I mean, we still count on you at the end of the morning.
Yes, yes, no worries.
And so now we are also happy to welcome Perrine Portier. Perrine is of course I know her because she's French, so it's much easy for me. And so she's the curator of INRAE CFBP and the culture collection from plant bacteria. She's a specialist in identification, characterization, phylogeny, taxonomy, diversity of plant-associated bacteria. And today she will present us the result of a project aiming at evaluating the viability of plant microbiota for different modalities of preservation. So please, Perrine, the floor is yours.
Okay, thank you, Jean-Luc. I will share my screen now, and I think it should work. Can you see it? Yes. Okay. So hello, everybody. I will talk about our project, which aims at evaluating the BAs induced by preservation. Well, I had planned quite a long introduction, but actually Thomas Ryan just explained everything that I wanted to say. So maybe I'll go a bit quicker on that part, and I will present maybe a bit more about the collection itself. Well, plants, they do not live alone, and exactly like humans, for instance, and they have a big microbiome, and which can be involved in biomass production, flowering rate, drug tolerance, disease resistance. And here in Angers, in IFHS, and more precisely in the Emergis team, we're studying the impact of microbiota during seedlings, for instance, and also we also study the impact of microbiota on the seed transmission of plant pathogenic microorganisms. And today we can see a lot of correlations because we can measure with different microbiota. For instance, the flowering rate of plants is different. Those microbiota on seedlings can vary along the time, etc. But now what we need to do is to go beyond these correlations and to assess the causes of the what happened on the plants and between plants and microbiota. And one way to answer that is to go through synthetic community studies. And these syncoms are formed by microorganisms isolated from these microbiota. And that can pose a problem, actually, because when you try to isolate bacteria from plants, there is only 4 phyla which are overrepresented, but only 4 among all the ones which are present. Okay, these ones are the most present in the plant microbiota, but it's not enough. And this poses the question of the syncom representativity. Another way to address this question and to try to find the real impact of microbiota on plants and to try to manipulate this microbiota to improve plant health is to inoculate native communities on plants and followed by phenotypic evaluation. And this is already done in human, for instance, with fecal transplantation. And we have less control on the community, but actually the community is transferred like that. Are closer to the natural ones. They are more diverse and more resilient and can have a lot of different interests. But these arise the question of the preservation of these native communities. Actually, if you want to use them on different plants, you have to preserve them. And I think the presentation of Matthew Ryan was perfectly illustrative on that. So thank you, Matthew, because that's my introduction. And I've looked in the literature, and for plant-associated microbiota, actually there is mostly no studies on how you can preserve them. So maybe there will be well, not maybe, but there's new papers arriving, obviously, but well, they were not available lately. Well, so a plant community is formed with bacteria, fungi, and protists, and if you want to preserve them, this will have an impact on the taxonomic composition because not all of them will survive. And in turn, this impact on taxonomic composition will have an impact on the functional diversity. So we decided to measure these 2 aspects of the community to be able to evaluate BAs induced by the presentation. And we did that in a project called MicroStore, funded by INRAE. And contrary to what Matthew presented, it's a really small project. And we did that at CMCFPP because, as Jean-Luc said, I'm the director of this collection, and our missions are the missions of all collections, meaning that we preserve the resources. Here it's plant-associated bacteria, mostly plant pathogenic bacteria, I must say, with associated data. We distribute them to the whole scientific community, and we valorized them also. This collection was funded in the early 70s here in Angers, on the base of the big collection of the researcher here. And from the beginning, they wanted to have a really well maintained collection. And now, almost 50 years later, we have a collection. We have about 7,000 accessions. We are really focused on the plant-associated bacteria. We are certified ISO 901. We are part of the SIM network. And like Dsmz, we are also a registered collection for everything which is related to the Nagoya Protocol. And inside this collection, we are specialized in the preserving isolated strains, plant-associated strains, and we transposed these techniques directly to communities. So these techniques are lyophilization. So the bacteria are first dried and preserved under vacuum in glass vials here, or cryopreserve them either at -80 in a deep freezer or at -196 degrees in liquid nitrogen. And the principle of our project is fairly simple. We took a plant sample and we focused only on radish, and we took seeds and leaves from plants grown from these same seeds. And we also constructed a little synthetic community from strains isolated from radish mainly. And we extracted this microbiota, and either we analyzed them directly or either we added a cultivation stage, either in liquid or solid medium. And after extraction, we analyzed first the taxonomic composition, meaning that we extracted DNA and we run metabarcoding analysis on 3 different markers, 16S and GB for bacteria and ITS1 for fungi. And we also assessed in parallel the functional diversity, and we used the biolog approach. We used their echo plates, which are designed specifically to make the metabolic fingerprint of communities. And they are formed by 31 carbon sources in triplicate on the same plates. And we also, from the extracted microbiota, we put them in preservation. And 2 years after, actually, this was initially planned in spring 2020, but it was postponed in 2021. And we took them out from preservation. And here also, we decided to analyze them after directly or after cultivation stage. This stage can be really necessary if you want to multiplicate your microbiota, to use it at a large scale. And after preservation, we also analyze taxonomic composition and functional diversity. Okay, so first, a small overview of the result before preservation. So as we always, that was not a surprise, but the diversity is much more higher. It's much more diverse. The microbiota are much more diverse in the leaves than in the seeds. And after cultivation, the diversity decreases dramatically. And that was expected too, because we can't have access to the whole diversity while cultivating. And this can be seen for the 3 different markers. That's applies to bacteria and fungi. We also compared the different strains forming the synthetic community, this strain alone here, and the metabolic fingerprint of the whole population here, and we did in duplicate, and we did not detect emergence of new metabolic capacities, neither we detected inhibition between strains. And when we look at the different microbiota, contrary to taxonomic diversity, there is here no influence of a cultivation stage, but we have a huge influence of the origin of microbiota. We can see that the different microbiota, they are clustered by their biological origin, actually. And after preservation, what we can see, it's here, we can still see more diversity in leaves than in the other different compartments. But we're not able to detect an effect of the preservation mode, neither an effect of the cultivation stage, and that I think that maybe the persistence of DNA in the medium can explain this, but we need to go more deeper with that. We also looked at the functional diversity, and here we're able to see, at least in leaves, that the preservation mode has an effect and can explain a part of the observable variation. Looking a bit more deeply in that, here you've got the measure of the intensity of metabolism. It's not the number of components which can be metabolites, but the level of metabolism we can see. And we can see that after preservation and liquid nitrogen, the maintenance of the metabolic function is better than compared to the two other preservation systems. And when we look at the cultivation stage, here you've got the seeds and here the leaves. And we can see that the metabolic intensity increases or is maintained after cultivation stage. This can be explained by the fact that the community, the organisms are growing in the media. And we were not able to detect loss of metabolic functions with the cultivation stage. So finally, these results are still preliminary. We're still in the process of analyzing them, and would like to determine more precisely the impact on the different taxa and the different compounds. But I think you will have to come back in 6 months, maybe, with the whole picture. And we can say that the taxonomic composition is not affected by preservation, but maybe because of DNA persistence. And it's contrary to what I wrote, so maybe I need to go back to my presentation, but the cultivation stage, at least in the beginning, they can have effects. And we've been able to see variations in the proportion of taxa. And on the functional diversity, it's affected by preservation, but not by the cultivation stage, and maybe because of functional redundancy. So after this work, we're able to have, well, not exactly now, but soon we will have precise measure of the effects. And we knew the preservation would have an effect on the microbiota, and now we have elements to measure them. And we can see this real difference between taxonomic composition and functional diversity, I will advise, if you want to really have a view of what preservation does on your microbiota, it's maybe more pertinent, more relevant to look at the functional diversity than the taxonomic composition. Well, and I thank you, all of you, for your attention.
Thank you, Perrine, for this nice presentation. And that really makes me wonder about how to preserve this microbiota. So,
well, that's a good question. Actually, I was really interested by the presentation of Matthew Ryan, because
exactly, we're all waiting for his results. Yeah. So, Francisco, do we have new questions?
Not as yet. So I invite everyone that wants to ask any question to include it in the questions option. And if we don't reply it now, we'll reply it in the question and answer session.
I have one small question for Perrine. Just how do you collect your microphotos between the seeds and between how do you collect your microbiota? How do you separate it from the seeds? Do you wash them in centrifuge, or because it's also a very huge step? to get it
Yes.
transformed to your
Our colleagues worked on the seed microbiota and how best to extract them. And it depends actually on the question you have. If you want to look at the microbiota, which is embedded inside the seed, for instance, or not, here we wanted to have, we wanted to measure the effect on the microbiota and not the effect of the microbiota extraction itself. So we took the simplest protocol of our colleagues. It meant that the seeds are soaked in water, or no, not in water, sorry, in buffer for 2 hours. And after we take this water, this buffer, and we centrifuge it. And we resuspend it in buffer, and that's the microbiota we work with. And for leaves, we crush the leaves in plastic bags with a bit of buffer too, and that permit the bacteria to go out. That's what we wanted to just extract microbiota directly. However, following your question, precise question, you may use different protocols for extraction. But I think these have an impact of what you can find after in your microbiota, but not on the preservation itself afterwards.
I think that, yes, meanwhile, we have a new question, but I think now it's, if I want, unless we can keep it, how much is it? If I know to have, I will keep always a 5 minutes switch between from the early timetable because of the difficulties we had at the very beginning. So the question that somebody asked was about how are we going to publish standard operating procedures? About isolation and the handling of microbes, and can we share them? I can say one thing is that for MIRRI side, we plan to publish standard operating procedures for the characterization and isolation of microbes. So it will be in the future. We plan to put that on the MIRRI website. Going back to Perrine, I suppose that if you're okay, that you are going to publish these results and all these protocols will be available one day.
Of course, we plan to make a publication next year about the results of our project.
Okay. So I'm sorry to have taken the liberty to answer directly, but it was just to try to keep on time. So thank you, Perrine, and we are now moving to something which is not exact. Well, it's complementary to the microbiota. It's the tools to explore the microorganisms that present this microbiota. So I'm really very pleased to welcome Professor Hauke Schmidt. And Professor Hauke Schmidt is professor at Wageningen University and holds a personal chair in complex microbial ecosystem. He's as well editor-in-chief of Dsmz Communications and senior editor of Microbiome and Animal Microbiome. Since 2018, he's the coordinator of the Unlock Research Infrastructure for research on microbial communities. And now he will present this unique new infrastructure. So please, Hauke, the floor is yours.
Thank you very much for your very kind introduction, and also thanks a lot for having me in this wonderful session. I think I can only agree with previous speakers how important and wonderful these initiatives like MIRRI, but also the microbiome support community are to get people together that are excited about microbiomes and microorganisms and discuss all those issues like the ones we've been discussing this morning. So what I want to do in the following is to introduce Unlock, something we are very passionate about, and I think which is very much in line also with the initiatives like the ones Matthew has been introducing in the first talk. So Unlock is an integrated infrastructure that combines what we refer to as biodiscovery, a biodiscovery platform, bioreactor facilities, and a data platform to really unlock microbial diversity for all the different applications and issues that society is concerned about. So really, I mean, if we think about it, where microbial diversity lies, then evolution of the past 3.5 billion years has resulted in this amazing diversity of microbes that's so incredible and endless that it's been often compared to the number of stars in the universe. But why would we care? And of course, we've heard a lot of reasons for that already this morning. And if we just think about ourselves, we cannot live without the microbes that colonize our own body at different places. And this is just one of the many examples. The plant microbiomes are other examples that we heard about this morning: chicken, pigs, to just name a few. And in all these places, microbial communities play essential roles in our daily lives and in the daily lives of animals and plants, for example. But I guess we can agree that this beautiful complexity of microbial communities is also the main reason why we still know so little about how they work, how they tick, and how we can get them to help us solve societal issues. And this complexity is probably also one of the most important reasons why industrial biotechnology, as we know it, at least until recently, almost exclusively relies on single microbial isolates. And these are, of course, wonderful tools. But these single isolated microorganisms probably comprise about 1% or much less of that of the natural diversity that we could be able to tap into. These single strain applications also don't take into account the opportunities and also the challenges that lie in the interactions that are normally present in microbial communities. But it's exactly these interactions that determine the function of these complex communities, and understanding these interactions is essential if we want to put these microbiomes to work for biotechnology and also for improving our health. And probably the lack of understanding these interactions is also one of the many reasons why so many microorganisms still remain uncultured in pure culture. So really, we need to step up to unlock the full power of microbiomes for a whole range of applications. So especially in Wageningen and Delft, I think over the past decades, we've built a lot of expertise to grow a range of different microbiomes in different types of bioreactors for different applications. Also in Wageningen, we are quite good at isolating or characterizing novel microbes, especially strictly anaerobic microbes from many different environments, wastewater treatment systems, the gut of different hosts. Also pioneering the use of all these omics tools to characterize microbial community function and composition. And also handling, us and others have been very active in developing tools to handle and use data according to FAIR principles. So what is it that we are really missing? Probably one of the most important reasons for that is the fragmentation of expertise and infrastructure. So what we aim to do with Unlock when we propose this to the Dutch Science Foundation is to provide or to provide the necessary integration, parallelization, and standardization of expertise, facilities, but also procedures that's so urgently needed to overcome these current limitations. And so about a year ago, we managed to convince the Dutch Science Foundation to support us with about 20 million for the next 10 years to build the Unlock infrastructure. And so Unlock comprises 2 bioreactor platforms located in Delft and Wageningen in the Netherlands. And you see that here, Delft at the western coast of the Netherlands, and Wageningen more or less in the center, closer to Germany. So 2 bioreactor platforms that provide the necessary hardware to grow and study microbial communities. Then the biodiscovery platform that provides facilities to, in an automated way and a highly standardized way, to process and analyze samples from these systems. And last but not least, also a data platform that then tightly integrates the wet lab platforms. So as I mentioned, Unlock officially started about a year ago when we received the funding from the Science Foundation as well as our 2 universities. And we are currently in the phase of building the infrastructure and the to be able to be fully operational, hopefully next year. So what does Unlock actually will bring us? So I mentioned already bioreactor platforms. And one of these platforms that is located in Delft will provide 40 standard bioreactors. And the idea about this massive parallelization of bioreactors is that if you want to test different microbiomes under different conditions, it will be very difficult to do that if you have to do that with 3 or 4 bioreactors like those that are normally available in laboratories. In a sequential fashion, then it will take us years to do these experiments. In a second platform that's located in Wageningen, we have a range of different complementary reactor types, standard bioreactors, but also bioelectrochemical systems, reactors to operate at high pressure and high temperature, as well as constructed wetlands. And those are more specifically geared to environmental biotechnological applications to treat different and valorize different waste streams. And we are currently in the process of building the laboratories and infrastructure to be able to place all these different reactors. Also in Wageningen, we have the biodiscovery unit. And one part of this biodiscovery unit is going to be systems to culture, especially anaerobes, at small scale. So starting from single cells, microcolonies, in different types of systems, suspended bead-based systems, but also culture chip systems. And to link that with microfluidic infrastructure that allows us to sort cells from communities based on different attributes, based on taxonomic attributes, based on functional attributes, for example, using Raman spectroscopy-based cell sorting, and then to handle these sorted fractions and cells, and to deposit or give them to the cultivation unit. In addition to that, we are going to build microbioreactor platforms that allow us to start screening, for example, synthetic mixed communities like the ones that previous speakers also alluded to. As I mentioned, we'll also have are also in the process of building a sample processing unit that allows us to extract different types of biomolecules, DNA, RNA, proteins, metabolites from samples from those experiments, and also then take subsamples into a cryopreservation unit to actually biobank material from those microbiomes. Also here in the middle of building these laboratories that are hopefully going to be fully operational somewhere early next year. Now a few words on the FAIR data platform. That's actually the most advanced in terms of development, because we had a little run-in time for a couple of years already. This FAIR data platform really drives on the, let's say, the need to be able to deal with data in a fair way. It's hosted at the national computing infrastructure at Surf-Sara. It's open source, and I guess it's also similar to for example the database platforms that Matt introduced to us before. So Also here, Unlock is a provenance-driven standard workflow, where we focus on having the data in a fair by design way. And what do we mean by that? That we take this let's say from the very start of the project, from project planning already, that experimental design and the, let's say, selection of metadata that should be collected are decided on upfront, and then data are being registered during the experiments, and then data are reposited and analyzed in a fair way. There's a number of aspects to that we are taking into account there. Standardizing metadata fields, for example, also using international standards, and more specifically, we are building that, or we built that based on an ISA project structure. So a data project structure where you take all these different elements from project investigation all the way to samples and assays into account to make sure that all the necessary metadata are actually being recorded and entered into the system. So as a user, you would then be dealing with a web-based interface where metadata can be registered and validated. And then there's standardized metadata fields, and there's restrictions on formats to make sure that metadata are actually compatible across experiments. There's standardized metadata fields that are also compatible with international standards, as I mentioned, because that's probably one of the most important limitations that us and others are facing, that meta-analyses are close to impossible, considering that metadata are not consistently recorded. Methods for wet lab experimentation are not standardized. So that's really, I think, one of the big challenges. In the web-based interface, you can select specific sample packages that allows you to sort of pre-select most relevant metadata. So for example, for the human gut or for air microbiomes, whatever you might be working on, and then add selected specific fields from an extended menu. Then those metadata are linked to the experimental data in a standardized workflow that's based on IRODS. And so in a nutshell, we think that this entire fair data framework will really allow us to also do meta-analyses and to link that across the platform. Obviously, this is open source, so everybody also unrelated to Unlock can make use of that, and we are already discussing with a number of different programs within and beyond our own universities to roll this database system or to make that available to others. So how can users actually make use of the Unlock facility? Well, it's basically an open procedure for transnational access, and users or interested scientists approach us with their ideas, and then together with our platform managers, we'll develop a project proposal that is evaluated by an independent external review board. Not huge proposals, but just to make sure that the science is sound and taking into account the fair data principles and fair experimentation principles. And basically, it will be possible to do experiments or to use Unlock at different levels. So you can use it as a basic, largely subsidized level. Of course, that's where we get the public funding for. And so we would help users with project design and operating the reactors, for example, and the other infrastructure elements, and train users on that, on the usage, but also on interpretation of data, for example. Then, of course, that there might be cases where users would want us to take actually care of the actual reactor operation, and that's possible. That might be a bit more expensive, obviously. And then, and this is a service, for example, to commercial entities, where we would be able to execute the full experimental setup, data processing up to a technical report. This will be only a small part of Unlock because we are really focusing on academic excellent science, but there is a possibility to use the infrastructure also for those more bilateral activities. Of course, you can meet Unlock already on the website. We have a newsletter that you can sign up for on our website. We started having webinars, blog posts, wide range of social media coverage, and also until we are actually up and running with our actual experimental setup, we try to already put up case studies that allow researchers to get an idea of what will be possible in terms of experimentation, but also data analysis on our platforms. The idea is to have an opening of the facilities in next year summer. Obviously, that might be restricted by restrictions that we're all facing currently, but that's the idea. This is actually not the building you see is actually not the Unlock facility, but it's the new conference center that's currently being built at Wageningen campus. So we hope that we'll be able to host that there. But again, especially the Fair Data platform, that's already fully operational and of course open to users. We organize educational elements, particularly for graduate students through our graduate schools, and also really like to see us as a hub for collaborations with MIRRI, but also IBISPA, for example, the industrial biotechnology accelerator. That's also a European infrastructure. And as I mentioned, we closely collaborate with the national computational infrastructure, SURF-SARA, in the Netherlands. And so with that, I'd like to thank you for listening to me, and of course I also want to indicate that I'm not doing this alone. So here you see the 4 platform managers heading the experimental and data platforms. And please, by all means, get in touch with us to discuss about Unlock and how we can collaborate to do all this wonderful science on microbiomes. Thanks.
Thank you, Hauke. It will be a very impressive facility or infrastructure and I hope that it will be launched very soon. So are there any questions, Francisco?
Not yet, John.
Well, just I have one question. Meanwhile, I have just one question that came to me because you spoke a lot about fair data, and you spoke about ontology. I was really wondering because technically, if you go to the microbial field, we can see today that there are, for some data that are physical data, that are not quite normalized, and so it's rather easy to cope with the fair and the compliance. However, if you look to things like habitats, microhabitats, and so on, there is a big requirement from ontologies, and so from the experience that we have at MIRRI, that right now we are trying to bring our database as fair as possible and in the frame of the EOSC project. One of the issues that we have is also the way to give you an example, the ontologies of habitats and so on. So it's really, I was really wondering, how far are you in the fair scope, and do you have developed or do you attach them to specific ontologies? And is there somewhere we could see the structure of your metadata and how we could well, the idea is to not to reinvent the wheel if possible.
But that also holds for us. So we are also, of course, we don't want to reinvent the wheel that others already have developed. And you're absolutely right. I think for some data types, the standardization is rather easy, and for others that might still be challenging. I mean, for bioreactor experimentation, that's probably much easier than for open field experiments, for example. Absolutely. But I would be very happy to share or discuss separately about that. I think that's really important to also make sure that these different because of course we are not the only one, like you indicated. So to align that across infrastructures and across programs.
Yeah, that's really key to the future exchange, collaboration, and at least the reuse of the data. But really, yes, it's something. So yeah, something. Do we have new questions now?
Not yet, look.
But people are too much impressed by the facility. Now we're longing to see how it can work. And so, and I have one question about the you focus more, let's say, more on the function of the microbiomes for, let's say, biological application, and much less than trying to disentangle the complexity of this microbiota and try to.
Yeah, I mean that's what I focused on today, maybe, but of course we are also in our own research very much interested in disentangling, for example, also assembly rules of intestinal microbiomes. How do they work? And that knowledge is essential if we want to actually influence microbiomes through early life nutrition, for example, or I mean, if we don't understand the ecological basis of how these microbiomes develop and are resilient or resistant to perturbations, then we will not be able to really influence them through nutrition or probiotics, for example. So that's definitely one of our main focus areas.
Okay. So I think, even though it's not, we should maybe switch on to the next speaker, and I hope that plenty of questions will appear meanwhile. And thank you again, Hauke. So now we are going to speak. We are going to welcome Dr. Vincent Thomas. Vincent Thomas is the head of the Microbiome Research Program at BioAster, Microbiology Institute in Paris. So BioAster is something new. It's a non-for-profit technological research institute dedicated to applied microbiology and infectious disease. That was created in 2012 by the French government. BioAster conducts innovative collaborative research co-funded by private partners and public funding in major application fields of microbiology, including microbiome research. So today, Vincent will speak us about targeted culturomics. Please, Vincent, the floor is yours.
Thank you, Jean-Luc, for the kind introduction, and thank you for giving me the opportunity to present this work today. So yeah, I will speak to you about flow cytometry for, let's say, targeted enrichment, maybe rather than culturomics, of complex ecosystems species. I will first start with the context. So here we mainly deal with we are mainly interested in the gut microbiome, but this because this is our subject. This is one of the main subjects we are investigating in the institute. But these tools that I will present can be used for many different other different microbiomes. Yeah, concerning the host-associated microbiota and the gut, mainly the gut microbiome, as you all know, there's an increasing interest with many potential applications that have been described these last few years, covering a very large variety of diseases, and different applications in the fields of, for instance, diagnostics, so the treatment of the disease or the prevention, drug discovery. So really, there's a very huge interest in this quite recent field of investigation. And this slide is to give several examples. This is not exhaustive. This is just to give several examples of companies that are trying to develop strains, so live biotherapeutic products that are single strains or cocktails of strains that will be used to treat or to try to prevent or some variety of diseases. So as you can see on the slide, that historically this kind of approach has been started to treat C. diff colitis. So this is in this field that you have the most advanced products, with products from serious companies, for instance. But there are now a huge variety of companies developing such products in other indications. This slide is to illustrate let's say the classical live biotherapeutic product discovery path. We were not at all involved in this work. This is just to give an example. So usually this discovery path, it goes from association data to active strain isolation. So for instance, in this paper that deal with Alzheimer's disease, the authors, they were able to demonstrate some differences in gut microbiota compositions between healthy patients and healthy volunteers, and patients. And so based on this, they tried to isolate the bacteria that were more present in healthy volunteers, with the goal to perform preclinical studies and identify strains that are more active than others. So what I want to illustrate here is that it's been a difficult process for us, because authors they needed to isolate and cultivate more than 4,000 bacterial isolates to be able to recover only 10 Faecalibacterium processes strains. So it does illustrate that it can be a very fastidious process to isolate specific species of interest from this very complex microbiota. Next one, sorry. This is just a very short introduction about the culturomics concept that's been introduced basically by the DJ Hall's team a few years ago, and which is quite simple. I mean, it means, at least in this definition, it means using a very huge variety of cultural conditions to be able to cultivate as many different species as possible from one same sample. We used this approach in past studies with our partners. As you can imagine, it can be very fastidious because you will have to use many different culture media, many different conditions, and antibiotics, etc. So it's very powerful, but it's very labor intensive. And this is non-targeted, meaning that you will be able to establish a collection that cover a variety of species, but this is not targeted. This is kind of random. So in this context, we choose to develop flow cytometry as a tool for culturomics. As you know, flow cytometry is a constantly evolving technology, and now we have flow cytometers that can detect very small particles with high resolution, and there's a constantly also increasing number of detectors that can be associated with the flow cytometers. So now, with the very last generation of cytometers, you can detect as many as 20 different channels in the same staining. And so as a consequence, flow cytometry is increasingly being used for microbial populations analysis. And there's an increasing number also of staining methods that are being investigated and validated for microbiology. So we were lucky enough to be in touch with Geert van den Eng, with the guy here on the left, and Jan Beyer from the CEA in Fontenay-aux-Roses. Geert van den Eng is a kind of pioneer in the field of flow cytometry. He really developed some of the very first flow cell sorters several dozen years ago, including the BD influx cell sorter. And Geert also is the co-author of this paper that's been published in 2015, describing the modification of the BD influx cell sorter by just plugging a glove box on the sorting chamber. And this glove box then is fed with nitrogen, constantly fed with nitrogen during the sorting process. And so the consequence is that the oxygen is completely depleted from the sorting chamber. So basically what we did is just to be in touch with Geert and to do the same thing with our BD influx cell sorter, and then apply this to gut microbiome studies. First thing was to demonstrate that we are able to preserve the variability of very oxygen-sensitive species during the sorting process. So for this, to achieve this demonstration, we used 3 different strains of the very oxygen-sensitive species Phytobiome prosnisi. And so here you can really see that if you perform the sorting in normal atmospheric conditions, the bacteria are completely killed. This was done without any additional staining. This is just sorting of the bacteria without any staining. And if you do the sorting under anaerobic conditions, then you can recover the bacteria, the colonies, by just putting 1 bacteria per spot, you get 1 colony. So there's a very good variability. Then we also were interested in validating the very classical live dead staining methods that are used for many different applications. But in this case, since we were lucky to have this flow cell sorter under anaerobic conditions, we wanted to validate the live dead staining with strictly anaerobic common cell species. And here, by sorting the live and the dead fractions, we were able to demonstrate that there's around 1.5 to 2 log differences between the number of bacteria you can cultivate from the live versus from the dead fractions. And it's been done for a variety of common cell species. So that was a good point. Another thing we are doing so these are tools we are developing to enrich specific species. So here again, this is very simple. These are we generate polyclonal antibodies by injecting rabbits with heat-inactivated bacteria. And so by doing this, we established a collection of polyclonal antibodies directed against beneficial common cell species such as Akkermansia mucinifera and the various Phytobiome species. But this is not exhaustive on this slide. So we have now a collection of, let's say, maybe 10 different polyclonal antibodies that target 10 different species. And we also have antibodies directed against detrimental gut common cell species, such as, for instance, Fusobacterium nucleatum, that has been associated with colorectal cancer. Another thing we are investigating is the reverse genomics approach, because as you know, many of these gut microbiota species. They've not been cultivated so far. So you cannot use this eat-inactivated bacteria approach because there are no isolate available in the collections. So in this case, what you can do is to investigate the genomes of the bacteria to identify to identify genes that encode for proteins that are supposed to be expressed at the surface of the bacteria. So they're supposed to be immunogenic. And then express these proteins in E. coli, and then use the purified proteins to vaccinate the rabbits. So there's been this very nice paper published by Mercier et Podar 2 years ago, and this is also an approach we are investigating, and actually it works quite well. I mean, you can do this, and it works. We are able to demonstrate it works. Another step in these developments was to demonstrate that labeling, staining, has no detrimental effect on the cultivability of the bacteria. So we were able to demonstrate that, for instance, the dye which is used to stain all the bacteria in the live dead staining kit, so the Cyto9, has no impact on cultivability. And if you perform the live plus the antibodies staining, here again, there's no dramatic impact on the cultivability of the bacteria. So this is a kind of overview of the pipeline. So we dilute the fecal samples in PBS, and then this is filtrated to avoid clumping of the sample lines. And then you can do the staining. So usually we use polyclonal antibodies in association with the live dead staining to be able to focus on the bacteria that are supposed to be alive. And we also investigate the use of additional staining, such as, for instance, lectins, such as the WGA. Once the staining is done, the sample is covered with paraffin oil to avoid oxygen exposure when the sample is taken out of the anaerobic chamber. And then it's plugged on the flow cell sorter to do the sorting under anaerobic conditions. And then the plates are re-incubated in the anaerobic chamber. And then once you have the colonies available on the plate, you can do the colony picking, and we use species-specific PCR to screen the colonies. And we also use typing, very simple typing methods. We use a random amplified polymorphic DNA because we could observe that for many samples, you can isolate several different strains of the same species of interest from the same sample. So in order to avoid to select only one strain, we try to do some random, some molecular typing methods with the colonies. And then we perform classical identification using 16S sequencing or even full genome sequencing. So this is the first proof of concept study we did with here again with the very oxygen-sensitive species Faecalibacterium prosnitzi. So this one is very oxygen-sensitive, but it's still present in relatively high abundance in the fecal samples, since it's supposed to represent 5 to 10 or even 15% of the gut microbiome. So it's not really difficult to detect. So here we use two different polyclonal antibodies. One that was generated with the Faecalibacterium prosnitzi A2165 that belongs to phylogroup 2A. And the other polyclonal antibodies was generated with a mix of these two ATCC strains that are very close to each other. And so what you can see is that the first antibody it does react with the A2165 strain, whereas it doesn't react as well with the two ATCC strains. There's much less staining. Whereas with the antibody that was generated with the two ATCC strains, it does react very well with these two strains, and it reacts only very, very slightly with the other strain. So then what we did is to use, we used a mix of fecal samples that were collected from 2 healthy volunteers, and we stained these samples with the 2 antibodies, and then the stained fractions were sorted using the flow cell sorter, and we sequenced the sorted fractions. So as you can see in the original samples, mix of samples, the fecal bacterium prosody is less than 5% of the population, whereas in the sorted fractions, there's a dramatic enrichment with the targeted species that does correspond to the phylo groups for which the antibodies were generated. And so then the next step was to use this method to isolate and cultivate new fecal bacterium prosody strains from fecal samples that were collected from healthy volunteers. So this is just to show the strategy. So we collect the fecal samples, and then we perform the live dead staining. And then among the live bacteria, we focus on the bacteria that are labeled with that are stained with the specific antibodies, and then we do the sorting and cultivation. And so using this approach, we were able to generate very quickly a large collection of fecal bacterium prosody strains that cover the phylogenetic 3, so a large diversity of strains. The second proof of concept we did was with another species, which is Chrysanthenella minuta. This one is not so sensitive to oxygen, so you can expose to oxygen for several minutes. It will still survive quite well, but this one is contrary to fecal bacterium prosody. This one is very low abundant in the gut microbiome, since it represents less than 0.5% of the gut microbiome. And interestingly, it's been associated with a lean phenotype. So it seems that this could be a good candidate to develop next generation probiotics to try to try to limit obesity. So there are companies trying to develop next generation probiotics based on these Chrysanthenella minuta species. An interesting point is that there's been this very interesting paper published 2 years ago, in which authors they built a very large collection of strains from gut microbiome that were collected from 11 donors. And using a large variety of cultivation conditions. And here they were able to cultivate around 8,000 isolates. But as you can see, there was no Chrysanthenella in this collection. So it means that if you don't use this kind of if you don't use a kind of enrichment method, you are not very likely to cultivate these low abundance species. So here, this is the same strategy we used. So we generated polyclonal antibodies that were directed against Chrysanthenella minuta Dsmz strain. So as you can see on the slide, the antibodies, the polyclonal antibodies here again, they were quite reactive. Again, they presented good reactivity against the strain. Whereas they presented only limited reactivity against other Chrysanthenella species, Chrysanthenella massilensis and monensis. And here again, we wanted to check if there is no cross reactivity with other commensal species. So we use a mix of fecal samples collected from healthy volunteers, and in this case we had to spike the samples because the target species are present in so low amounts that you will have to sort the bacteria for hours and hours before having enough bacteria to do the sequencing. So the samples were spiked with the pure culture bacteria, and then as in the previous study, we sorted the bacteria, and we sequenced the sorted fraction. And as you can see, there was a very good enrichment, but also some kind of cross-reactivity, since there were a couple of commensal species that were also enriched. Sorry. And so using this strategy, in this case, in addition to the variability staining, so we, as I explained, we do the variability staining and obviously antibody staining, but in this case, we also use the size criterion as a selection criterion, because the Crescentina minuta, it means the minuta means it's a very small bacteria. So we focused in the dot plot on the region in which these very small bacteria were abundant. So we took there were three criteria. So the live bacteria, small bacteria, and that are stained with the antibodies. And then we were able to demonstrate by doing some cultivation experiments that by spiking with as low as 0.1% or 0.01% and focusing on this region, so live, small, and stained with the antibodies, we were able to cultivate more than 90% of the isolates that corresponded to Crescentina minuta. So then, using these tools and this approach, we were able to very quickly build a collection of strains that are now being used by the company Isopia Bioscience to develop next generation probiotics. So now this is the conclusion slide. So I hope I demonstrated, and I hope I convinced you that the flow cytometry is a versatile tool with many potential applications in the field of microbiome research, from host associated, because this is our focus, but also for environmental microbiomes. And then I will also mention, since Jean-Luc mentioned Biodiversity is quite recent, so it's a private, non-for-profit research organization, and when we are very interested in collaborations to work on complex microbiota analysis using flow cytometry, as well as selection of specific microbial targets from any complex ecosystem. So we use flow cytometry for the analysis. We also use for the analysis and the monitoring of microbiota, and for the targeted enrichment of specific species. So feel free to contact us if you have any idea or collaboration project. And with that, I will finish. And these are the people we collaborated with for this project. So people from Gervon and Eng and Jan Bayer, and also people from Mat Pharma, which is a French company developing fecal microbiome transplantation. Isopia Bioscience that develops Crescentina minuta as a next generation probiotic. We also worked with Stomach Level in Aachen to do the sorting and sequencing experiments. And that's it. Thank you.
Thank you very much, Vincent, for this very nice talk with the strategy that I do appreciate quite much. So I'm gonna ask to Francisco if we have questions.
Yes, we do, Jean-Luc. First, we have one specific for Vincent, which is from John Swings. The question is, are there research groups using culturomics for the treatment of autism?
I know for sure that there are research groups interested in the possible links between autism and gut microbiome composition, and so I guess there must be some groups trying to balance gut microbiome composition to try to help alleviate autism symptoms. There must be. I'm sure there are, because there are groups working in so many different applications in the field of microbiome today. But I cannot tell you which bacterial species they are interested in.
Thank you very much, Vincent. Now we have 2 more questions connected from our colleague Raquel, but this is actually for all speakers. So I will read the questions. There are people interested in preserving samples of a mix containing microorganisms and human or animal cells. Do you consider these mixed populations as microbiome? And do you consider that this could be an important need for culture collections to preserve that kind of samples? So maybe Jean-Luc, I don't know if you want to comment on this first or then pass it to any of the speakers.
Technically, this is one of the main question of the discussion that should have right now, and technically that we are arriving. So it's a pity because we are squeezing a little bit the question that may arrive to Vincent, but these are broad questions about that, about the importance, about the role of culture collection to preserve, to collect microbiomes and or strains from the collection. I remember something that from Emmanuel Maguin, she was making difference between microbiome and microbiota. Microbiota is only the set, let's say, of microorganisms, and microbiome includes all the environmental aspect. Please correct me if I am wrong. So technically, I would say I would think more about microbiota than if I'm correct. No, but please, you are the specialist, so please. Who wants to take the floor?
Well, maybe I can start. Your definition of microbiome versus microbiota, I think, is completely right. And so the microbiome would also include animal cells or human cells. I guess the question is whether the same preservation methods would be appropriate for the different types of cells. And that is probably the biggest challenge. And so yeah, I think it's important to, especially if you think about the individual or personalized microbiomes that individual humans have. And I think it would be important to have biobanks of let's say, connected tissue, human cells and microbiomes. But whether that would be one sample or let's say a connected parallel biobank with different cell types, I guess that's more a technical question than a conceptual one. But it's definitely going to be important, especially if you think about personalized medicine, to have those tissues and cell types connected.
Any other comment from other? But for plants, from the plant side?
Yeah, I think Emmanuel is exactly right with that description, and you do need to make it. I think microbiome also includes, from a preservation perspective, anything that's unculturable. And I think that's a key difference. So you've got the culture-dependent approaches and the culture-independent approaches. And yeah, but the methodology, as Hawk said, is part of the challenge we have. The methods need to evolve. And actually, Perrine's brilliant talk, she's found something similar to we have, in that liquid nitrogen preservation actually seems to do a better job in preserving microbiome type samples. And that's important because many scientists throughout the world distribute samples without due care and attention as to how they're getting them from A to B. So it really is an important aspect that we as preservation scientists and culture collection people have to address in order to make sure that the quality of science is what it says it is. Because I suspect that if you take different samples, the same samples, you treat them differently and do the same study again, you'll end up with different results. And that's quite concerning.
Well, in microbiota, even if you take the same sample and treat it in the same way, you can have different results. That's another problem. Well, I'm not competent in the preservation of plant cells, but I have colleagues who tried, and it's much more difficult than preservation of microbes, so it can be difficult. However, we have a question here in our lab. We work mainly on a seed microbiota, and maybe the best way to preserve the native community is to preserve the seed, actually, because when the seed forms itself, it's real dry, and the microbiota adapts to this new form. And actually, the microbiota on seedlings is really radically different, because the conditions are different. But it can be a solution, but in some cases only, and for the other, that won't work, actually. So I'll.
But the presentation of Matthew, thank you, Perrine. I was also mentioning that there was the threat of preserving seeds and not having the whole microbiota as well, and especially if you do long-term preservations of seeds. We have these gene banks of seeds, and on short term, I'm pretty sure that we do quite well with the seeds, because basically this is the natural biological cycle. But on those long-term preservation in place for seeds, do we have the same insurances?
Maybe yes, because in fact the critical steps for seed formation, it's when the seed itself dries out.
Mm-hmm.
And at that point, a lot of things happened. And after, it's like in a stasis. So I don't know if we can preserve them for 30 years, maybe, but for 3-4 years, the microbiota survives on the seeds. But as I said, it's a very specific microbiota, and the microbes inside are in a resistant form. So it can answer some questions, but not all of them. And if you want to work on the leaves microbiota or roots microbiota, that won't work, because the leaf will decay with time and quite rapidly, especially for the annual plants and so on.
Okay.
There's actually a paper published by the Millennium Seed Bank at Kew Gardens by a researcher called Rowena Hill that addresses some of these issues. And they found that there was only a percentage of their seeds where they were able to isolate fungi from. So it's a really good paper. So if you can access that, please do, because it really emphasizes the fact that, yes, seeds are a good mechanism. And actually, going back many years for European-based research from work done at the Westerdyke Institute by Hugh Stelpers and Shui Tan and David Smith, they were actually using seeds as a mechanism to preserve recalcitrant oomycete fungi. Well, they're not really fungi, as we know, but the seeds provided a protective mechanism. But with all of these things, when you're dealing with populations, there will always be a proportion of them that aren't very freeze tolerant. So you have to ask yourself, are you actually selecting the components of your consortia that are freeze tolerant? So there's lots of work still to be done. And this is where, as a community, we need to bring our research together to start asking some of these really fundamental questions.
Yeah. Do you have new questions, Francisco?
Not new ones, but I want to revisit one from Lucille that made a question back in Matthew's presentation. Of course, if any other speaker would like to comment, of course, they're welcome. But the question to Matthew was, if you have some ideas to better manage and share collection samples with new AI and digital solutions.
I have no idea about artificial intelligence, and I defer to anybody on this panel who knows more than me about it.
But maybe Hauke might have ideas about it.
Well, I get them with Matthew that I really don't know much about that. Just that that there is increasing promising, well, that people think that these methods really have promise, bring promise to these big data and to provide provenance of data. So I think, yeah, that's definitely something to take up, especially when we look at these complex microbiomes and to be able to store and interpret the complex data that are associated with that. I mean, just the discussion we had right now that on developing plant microbiomes develop along the germination of seeds, and the same, of course, in invertebrates and other animals. And we will not be able to biobank all the physical biological samples, but we also have to think about connecting and biobanking the data that go with these developmental programs. And I guess for that, artificial intelligence is extremely important to make sense out of that data.
Okay, thank you, Hauke. I've forgotten. I had one question to Thomas that I have forgotten to ask him at the end of his talk. Is that in your efforts to characterize as much as possible the mechanism of the microbiota of the microbiome of pigs, pigs, mouses, and chicken. And as far as I remember, my question is, how many genus do you have in comparison? Do you have isolated in comparison to those that have been characterized through metagenomics studies? And so my second question would be: do you plan to explore with specific tools, like something like the approaches of Vincent or something like that?
This is a good question. So we are also active in metagenomic or the metagenomics. So on another project, we are sequencing the complete metagenome of fecal samples and try to isolate the samples from this fecal, fecal isolation. So we will find, or we are trying to find, everything in single isolates, which we sequenced with the metagenome. So this is another project where we are focusing on, so it's connected with. And I have, when I see the question from Raquel, I think, regarding the culture collections, or if we can preserve some samples or microbiome samples, so we are already active in that in form of artificial microbiomes. But I think it's a lot depending what you are looking for. For example, when you have to, how to say, I think the tissue microbiome is different than the bacterial microbiome or an artificial microbiome. And for the tissue microbiome, it's not easy. So you are having not the same, how to say, the same samples. You can not regenerate them because it's sampled, for example, from a patient. And you may not have been access to the same patient again. So you cannot revive the tissue with the microbiome. So that makes it really not simple to preserve it. And at the end, you will have not the same sample again. With bacterial samples, or artificial microbiomes, when you mix it, you have several problems. For example, when you wish to have a microbiome with, for example, 12 or 20 or more bacterial samples, you will have problems at first when the amount of bacterial strains is increasing. You have the amount of bacterial samples which you have to manually control. And since many microbiome gut samples are anaerobic, you have also a problem with in kind of robotics, I have to say, because many robotics do not work anaerobically. So you only be able to preserve, I think, aerobic or microaerophilic bacteria as microbiome. And the next thing is, I have to say, the bacteria have different growth behaviors. And when you wish to have a microbiome, for example, with a bacterium or with several bacteria which grow which grow, for example, the first bacterium 1 day, the second 2 days, the third 10 days, and the fourth 20 days. Then all aerobic or anaerobically, then you have a problem or you might have a problem in handling it en masse, for example, 40 bacteria together, depending alone on their growth behavior, independent of robotics. So you cannot mix 40 bacteria so with different growth behaviors. When you have to begin with the longest growth behavior, you have to begin with the bacterium which takes the longest time. And you have to end to cultivate with a bacterium which takes the shortest time, and then you mix all together. So this makes it really difficult, and I think 20 strains in a mass of an artificial microbiome independent of tissue microbiomes is the maximum which you can manually handle, I think. So this is my answer to this special question of Raquel, I think. So it's not easy to include tissue-related microbiomes or microbiomes in mass in culture collections. It's possible, but not in mass. It's my personal opinion. It might be that there are other opinions, but this is my opinion to that. So I hope this helps a little bit, or maybe there are other opinions to that.
Thank you. Sorry, thank you, Thomas. So there is somebody who wants to. So thank you. Yes, I understand that the more species you have, the more differences in the growth abilities leads to the more complexity, and I can understand that. So, and I was thinking the time was rushing, and this is why I wanted to. But we have one last question, maybe from Lucille. But before, if I ask one comment, because I saw you were just about to say something. So please, no.
No. I think I completely agree with Thomas. I mean, conserving these microbiomes as collections of single strains, that's challenging, especially considering that many are not yet in culture. But I guess there it's important to also biobank the native microbiomes, as a future resource, also for isolation when we get better at that. And maybe another remark on the uncultured majority that you also asked about. I think there these reverse genomic based methods that Vincent also alluded to, combined with flow cytometry and cell sorting, that can help a lot, I think, to target specific cells that we otherwise won't be able to get a hand on.
Yes. So for Vincent, it will be, I think, maybe our last question. As a bacteriophages has an important part of microbiota, how flow cytometry could be used and applied to support new drug development?
Yeah, it's not an easy question. But at this stage, the flow cytometry is still quite limited to detect directly phages. I think it's been used to detect very large viruses. I think Didier Raoult's team in Marseille, they used it to detect what is called giant viruses, So they were able to detect these giant viruses. And so there are also quite giant phages. So maybe for these specific phages, you could use flow cytometry, but for most of them, I guess it would still remain very difficult. But there are maybe other approaches in which you could. This is something we are thinking about. If you are interested in getting phages from a complex ecosystem, maybe you could incubate labeled bacteria in the ecosystem, and then sort the bacteria from the ecosystem with the hope that the bacteria will be infected by phages. So using this kind of strategy, maybe you can go to some kind of fishing expedition to recover specific phages from complex ecosystems. This is to develop. This is something that needs to be developed. Yeah. I hope it answered the question.
Well, if Lucille says so, but at least it shows that we still have work to do, and so that's a good point about it.
A lot of work. Yeah.
So I think now we should have a break, and I would like to thank you all for being available and to present your as well results, your let's say ideas about microbiota and culturomics, and that was really for me a wonderful experience to have you with us today, and I really thank you for being there. So I hope that all participants will follow us this afternoon after lunch. So after you log out, you will have to log again, and so. And yes, that's it. So I would like have to have a nice lunch. Okay, thank you all very much, and I hope to meet you all in real life once.
Thank you.
Bye.
Have a good lunch. Bye bye.
Bye bye.
See you later.
Bye.
See you later.
Thank you.